Welcome to Bacteria, Antibiotics, and the Power of Nature. Did you know all living things are made of cells? Not all cells are built the same, though. The simplest cells, called prokaryotic cells, are like open floor plan studios, and everything happens in one shared space. Bacteria are a great example. Eukaryotic cells, which make up plants, animals, fungi, and protists, are more like high tech office buildings. They have a special feature called the endomembrane system, a network of internal membranes that divides the cells into compartments or rooms where different jobs happen. This system includes the endoplasmic reticulum, the Golgi apparatus, and vesicles. These allow the cells to multitask and run processes more efficiently. For example, proteins are built in a rough ER, then packaged and shipped by the Golgi like an assembly line factory. Now, if we compare plant and animal cells, they share many structures: a nucleus, mitochondria, and that same endomembrane system. But plant cells also have a rigid wall for structure, chloroplast for photosynthesis, and a large vacuole for storage. Animal cells don't have these, making them more flexible and equipped with lysosomes to break down waste. Now, when scientists design medicines, they're looking for something specific in a cell to aim at. We call this a drug target. A drug target is a structure or molecule in a cell that a medicine can attach to or affect in order to change how the cell works. Think of it like a lock that the drug's key can fit into. Even though prokaryotic cells and eukaryotic cells are very different, they share some common features that can be drug targets. Two examples are ribosomes and cell membranes. Ribosomes are protein factories. Some antibiotics bind to bacterial ribosomes to stop them from making proteins. While for cell membranes, the protective barrier around every cell, some drugs can damage these membranes, which can kill the cell. The challenge is designing drugs that attack harmful cells without without harming healthy ones. Now, antibiotics are especially effective because bacteria have unique structures that our cells don't. One example is the peptidoglycan cell wall. Human cells don't have it, but bacteria do. Penicillin blocks the enzymes that build this wall, weakening bacteria until they burst. Another is the difference in ribosomes. Bacterial ribosomes are smaller, 70S instead of our 80S ribosomes. Antibiotics like tetracyclines and euromycin can block these bacterial ribosomes without affecting ours. A third example is DNA glycase, an enzyme bacteria need to coil and copy their DNA. Quinolone antibiotics like ciprofloxacin stop this enzyme, preventing bacteria from multiplying. By aiming at these bacterial only features, antibiotics stop infections while keeping our cells safe. And now, the journey of an antibiotic. So, what happens when you actually take in an antibiotic? When you swallow a pill, it dissolves in your stomach and small intestine. From there, drug molecules are absorbed into bloodstream and carried throughout your body to the site of infection. Some antibiotics stay outside of cells, while others enter certain cells to reach hidden bacteria. How they get in depends on the drug. Some slip through membranes by passive transport, while others require active transport where energy is used to pull the drug in. Now, will any of your cells be killed? Usually, no, because antibiotics target bacterial features, not human ones. But sometimes side effects happen, since our mitochondria are distant relatives of bacteria. And antibiotics don't just kill harmful bacteria, they can also affect your microbiome, the helpful bacteria in your gut, mouth, and skin. That's why antibiotics can cause stomach upsets or other side effects until your microbiome recovers. Now, in conclusion, why it matters. Antibiotics work because they can target bacteria without harming most of our own cells. They aim at unique bacterial structures like cell walls, ribosomes, and enzymes. But bacteria are constantly evolving and resistance is rising, meaning today's antibiotics might not work tomorrow. Most of the antibiotics we use came from nature, from some soil, bacteria, fungi, and others from other organisms living in forests and ecosystems. And if we lose these natural areas, we may lose the chance to discover the next life saving medicine. Protecting biodiversity isn't just about saving wildlife, it's about protecting human health too. By preserving the natural world, we protect one of our most powerful resources, the potential to discover new cures.